we truly believe that biology is technology. It's just technology so advanced that no human will ever build it.
We decided to take two of the hardest things possible, biotechnology and semiconductor, mix them together, raise no money, and figure out how we're going to make this work, which sounds like a recipe for disaster. Turns out it worked, and putting those two together allowed us to really conceive of the technology now as a biosignal processing unit, something that directly integrates biology and digital systems to create a new type of technology that no one's ever been able to see.
We're taking the strength of modern digital electronics and applying that to really integrated biological components.
There's a number of measurements you cannot do today. If you want to know how an RNA binds to a protein, people look at you and say, well, unfortunately, you can either choose to look at the RNA or you can look at the protein. But that in-between state, because we only have sample preparation methodologies, as it's called, detection methodologies, that can do one at a time, you can't get the interplay between those two worlds.
When we look at the traditional mid-20th century methods used to understand biology today, we're extracting from a cell one element at a time, one protein, one set of nucleic acids, one small molecule, and trying to understand that one thing or couple things as a snapshot.
What is needed is data streams, video instead of pictures, if you will, because there's so much more knowledge and understanding if you see things developing over time. And because we have a technology where the one side becomes a part of the electronics, the other side, by definition, is the signal. That's how we bridge the biological networks and the digital networks.
For 40, maybe 50 years, people in electronics have been looking at incorporating biology into what they're doing. The issue is that typical electronics don't work well in salt water. So if you take a silicon transistor and you stick it in the most benign biological fluid you could think of, it will oxidize. But if you want to do something like look at what an enzyme is doing in real time, or do multiomics, look at genetics and proteomics and metabolomics all at the same time, you need something that can do a field effect measurement and survive being put in salt water, or blood, or urine, or buffers, any of that kind of stuff.
Bridging the gap between the silicon-based transistors and BPU using graphene, a wonderful material that is provided to us by nature, which is biocompatible, which is faster in transferring these electronic signals, so allows us to look at biological event in real time, can really expand the applications of all of these electronic gadgets that we have around us as a tool that can now be used to monitor our health. The process of developing this is a very complex process. It's not only going from genomics to phenotypes to proteomics. There's all of these events that are happening simultaneously that are defining the pathology that is causing that disease. If you can really tap into these multiple different biological events in real time and look at the interplay between genomic, proteomics, epigenomics, you can really understand the disease processes much better.
Our role, instead of creating or re-engineering tiny robots, tiny systems, it's to work with biology, to use the technology that exists and figure out how to integrate that. We've got this concept of a BPU, which is Biosignal Processing Unit. And this is the core of the hardware that's required to run the platform. The platform is the hardware, the BPU, the reader that controls the BPU, which you can think of as the chipset, but also the wetware. These components that can have meaningful two-way communication with biological elements, and the software, the part that tells the reader what to do, interprets the signal, presents that information to the user.
So the range of application can really vary from agriculture to molecular testing for disease diagnostics to quality control applications for CRISPR-based therapies and many others. Powerful applications of a multi-omics BPU platform is the ability to perform liquid biopsies. Liquid biopsies are very important for cancer diagnostics. where we need to be able to monitor free circulating DNAs, proteins, and other molecules. Imagine if we can do that all within the same platform and monitor how these different biomarkers and the interplay between these different biomarkers play a role at the different stages of cancer development. We anchored the CRISPR within our BPU, which allowed us to actually search for our target sequences in real time.
We've obviously done a lot of work with CRISPR, and the simple way to think about it is every biologist now can be a gene editor who understands basic biochemistry. Now they can do gene editing, and now it opens up so many more potential applications and ideas to just about everyone who can use a pipette.
There are a wide range of applications for CRISPR chip. This application ranging from wanting to do a rapid molecular testing, for example, in terms of detection of SARS-CoV-2. The process is super lengthy, you still have to do PCR amplification, and it requires infrastructure. Imagine if you can do that in a handheld device using CRISPR, using biology as technology that could lead you to much faster response. In agriculture, for example, if a farmer wants to know if the seeds that they are planting is the right seed, they want to have a test that is rapid, that can give them a result in 20 minutes. CRISPR chip can offer a tool to monitor the efficiency of CRISPR editing before and after editing, to really look at the percentage of the cells that have been edited successfully.
Now, today, there are companies developing therapeutics around CRISPR. There's all sorts of potential around diagnostics. They all got to get it right. The BPU is able to look at the CRISPR process at every step through the entire application. So we came up with our own QC methodologies to make sure we're getting good outcomes for our partners. It was a great way to showcase not only what Cardea could do, but then also make an impact in this emerging field that has so much potential but still needs good quality control.
We also have developed a new version of CRISPR chip, which can detect single-point mutations. And as you know, 50% of human diseases are a result of single-point mutations. So this could be very powerful technology for early disease diagnostics. Using graphene to bridge the gap between silicon transistors and BPU, now we have a gateway to biology.